of some of your more evolved thoughts. Without further ado, I will share my screen and get into my presentation. It may go a little long. Manjula, if you don't mind, you can just rein me in when when things are going too far.
No, it doesn't matter.
Okay.
It doesn't matter.
And I'll try and keep it as concise as possible.
Okay.
Normally, what I do is I plan for a half-an-hour talk, and it goes on for 1 hour. So let's see how this one works. Can you all see my screen?
Yeah, it's visible.
Okay. All right. So that's me on the right. That is me in one of my best moods. So I normally don't smile that broadly. And thank you once again to all the organizing committee for inviting me to give this talk. Like I promised, I'll give a small background about what I'm going to talk about first. And I'll go a little slow here. And the idea of the next 2 slides is simply, irrespective of whichever background you come from, what subject you study, is to bring you all on the same page so that from here on, hopefully we can all stay together in what I say. All right, in this rather busy slide, the question really is, where is our DNA? Right, so we can start before that with what is our DNA? DNA is the genetic material that each of us has. All of us has close to 1 trillion cells, maybe more, in our body that go to comprise the various tissues that we have, the skin, the kidney, the liver, so on and so forth. And in each of these cells, barring the RBCs or the red blood cells, there is a nucleus. And in that nucleus is the genetic material called DNA, or deoxyribonucleic acid. Now, this has been studied for some time now. The discovery of the structure of the DNA has actually won the Nobel Prize back in the 1950s. So it's quite a famous molecule, and I won't introduce you beyond that to what it is. But suffice to say, DNA is comprised of 4 bases. So if I were to draw a computation analogy, DNA is the hard drive of your computer. Okay, and your computer basically understands code in binary, which means zeros and ones. DNA understands its code in a quaternary manner. So there are 4 bases, A, T, G, and C. I won't go over their extrapolations, but suffice to say these 4 bases in various permutations and combinations are organized in the nucleus of each cell, and they determine what makes you, you, and me. And for that matter, what makes an animal, right? It's the order in which these 4 bases occur that determine, in many ways, what your genetic blueprint is going to be. So it may be something as large as that you will be a human, to something as minute as, yes, you will have blue eyes or you will be 6 feet tall. And all that is coded in your DNA, and basically the order in which these 4 bases are organized. And DNA sequencing, which is a tool which I will be talking about extensively, is a combination of some lab work, biochemistry, some hardware and computation to decode what sequence these A, T, G, C bases actually occur in your DNA. And why, you may ask? Just sort of briefly, I'll tell you. It's because, like I said, it's the blueprint, and it can predict, certain phenotypes that you may have. If they're clinical in nature, they can also predict pathologies, right? So many of you who are not fully familiar with how this is organized in a cell, if you can see the mouse arrow here, the DNA in the nucleus is organized in the form of discrete structure called chromosomes. Okay, and there are 23 chromosomes, and so 23 from dad, 23 from mom, so you have a total of 46. As a result of having this DNA organized in this manner, at a sort of minute level, you'll see that they are wrapped tightly around certain very positively charged proteins called histone proteins that help package them into these discrete structures that then go and sit in your nucleus, right? But at the base of all these, funky diagrams that you see here are the bases A, T, G, C, organized in a double helical manner, right? So with that, getting that out of the way, I'll just go over some of the ground rules of molecular biology. It's called the central dogma of molecular biology, right? So this is a cell that you see here, and inside is the nucleus. And of course, you have DNA, that is genetic material. And as I mentioned earlier, you can equate DNA to the hard drive, right? Then there's a transactional molecule that the DNA actually becomes before it becomes its terminally differentiated form. And that transactional molecule is something called RNA, or ribonucleic acid. Okay, and this ribonucleic acid, I would draw the parallel to a software that you may have, or your RAM, right? So you have a RAM that's sort of a quick access to what genetic material your DNA has, and it serves many purposes, including specifically only translating certain portions of the DNA, and that becomes very important in determining the fate of a cell type, right? And finally, of course, that RNA gets translated in a process called translation into something called a protein, and that protein then goes on to make the structure of your cell, eventually of your tissues, your organs, and then, of course, your whole being. And that I could draw a computer parallel to a printer. Okay, so it's a kind of terminally differentiated place in which a message can get translated to in the end, right? So this transmission of information from the DNA to RNA to protein was what is referred to as the central dogma of molecular biology. And by and large, of course, there are exceptions because biology is full of exceptions. But by and large, the transaction of any cell in any cell is conducted in this manner, right? So the DNA serves as a template of which the cell makes RNA, and then that RNA gets amplified, and of course, proteins are made from that RNA. So these are the base. This is just the basics of what we're going to talk about now. And I thought it's good that we all get acclimatized with it. On this side, I'll basically tell you the transformation of DNA to RNA. That entire process up until translation is the field called genomics. Okay, so whenever I say genomics, I'm referring to any one of these two currencies, or either DNA or RNA. For the purpose of today's talk, to keep it simple, let's just visualize it as DNA. Right? There is a factor called epigenetics, that is methylation and other such features, acetylation, that takes place, but we won't get into that today. And of course, once the RNA is translated into a protein, then the field is called proteomics. And so some of the things that Dr. Manjula alluded to earlier today in terms of prediction of protein structure, so on and so forth, are dealt with primarily in proteomics. Okay, so you won't hear much about that from me today, but we can have another chat on that on another day. Another quick piece of trivia that's important to know is that the human, the size of the human genome is 6 billion base pairs. Okay, so because it's diploid, so it's 3 billion in a haploid cell. So just imagine ATGC repeated in a distinct and unique order 6 billion times. Okay, that's the size of the genome, and that's what's packaged into each nucleus of every cell in every tissue of every organ that you have. Now that you understand the scale of what we're dealing with, right, I'm going to deep dive straight into the next part of what I'd initially planned to talk about. Now, I can pause here for a second if anybody would like to raise a question, or shall I just carry on, Manjula? What do you think?
Yeah, actually, you carry on. They'll post their questions in the chat box.
Okay, I don't want to lose anybody.
We can actually.
Okay, I don't want to lose anybody here. Okay,
yeah, okay.
I hope we're all on the same page.
Yeah.
If we're not, don't worry. I'll keep referring back to this. You can always come back to it, right?
Okay.
So the title of my talk was Genomics and Disruption, and I think the best way to deal with these three words is to understand them independently. Okay, so genomics I told you a bit about, right? The DNA and the RNA. But what is disruption? Okay, so disruptive innovation, this you can get off Wikipedia. Okay, a disruptive innovation is an innovation that creates a new market and value network, and eventually disrupts an existing market and value network by displacing the established market leaders and alliances. Okay, I think it's many words to describe. Maybe I can tell you a small story. It's in the early 90s, and probably for a decade in the 90s, late 80s and early 90s, there was a company called Kodak. And in those days, many of you were not around then, but in those days, whenever we took a photograph, it was captured on a film, right? And that film was developed. And that technology for developing that film was something that was patented and made very accessible to people through this company called Kodak. And it was the single largest company in terms of market share as far as taking pictures and the whole field of photography was concerned. Then what happened was the digital era came in, and before you knew it, you could now have digital photographs. And now you and I mean, many of you probably don't even know what it's like to see photographs on film, right? And it was almost overnight as far as the size of that industry was concerned, where the digital technologies came in and displaced a pre-existing technology, okay? Another quick example I'll give you is, back in the late 1800s, the automobile had already been invented. So Carl F. Benz had already made his internal combustion engine, and people were making cars, right? But it was what I'd call a pre-disruptive era, where rich people had cars and nobody else had cars. Everybody else had horse-drawn vehicles or horse carriages, right? And then this company called Ford, which you're all familiar with, built the first assembly line factory for a particular car, right? And started mass producing this in a factory. As a result of this, the cost per car came down dramatically, and therefore the access to that technology skyrocketed. As a result of which, by the early 1920s, the automobile was called a disruptive technology because it had displaced an earlier technology, okay? So where I'm going to with genomics is how genomics is already a disruptive technology and how much potential it has to disrupt in other areas. And so the rest of this discussion will be how genomics has been used in order to disrupt the way we do agriculture, the way we do clinical sciences today, the way we do research today. And I hope that, I will deep dive into some of the details. So even if you get lost along the way, don't worry. You can just remember that the theme is eventually this technology is going to get back and replace a whole bunch of other technologies. And which is why you should know about it. And which is why I think it's also important to understand that it's a multidisciplinary area, right? So there's a lot of computational analytics involved. If you cannot do this without computational analytics, and that's something I'll talk about. You also have a lot of lab work, so wet work. So there's a lot of room for biologists to participate in this. And of course, in terms of new applications, there's a lot of room for creativity in applying it to novel areas. And I'll hope to touch on one or two people who did that. But before we get into the disruption of genomics, so the disruption caused by genomics, it's important to do a small little history lesson. Okay, so I'm going to describe to you what I'm referring to as the precursor for disruption, or the pre-disruptive era. Okay, and let me see. Yeah. So in 1990, the National Institute of Health decided to undertake a very ambitious project, right? The project was to arrange these 3 billion bases of the human genome in the correct order, right? Given the technologies that existed at that time, right, they had postulated that this project would take 15 years, and they allocated $3 billion, okay, in order to do this. And every year, as you can see in this time chart, every year little progresses were being made, and these were being sequenced one chromosome at a time. It was a multi-country effort. It was very international in nature. Unfortunately, India did not participate in this, although China and other countries did. And of course, to make a long story short, 15 years or 13 years later, that is 2 years ahead of schedule, And at a cost of $2.7 billion, which is again a cost that is better than what was predicted, the first human genome sequence was published. Okay, it was published by two groups. One was a private group, a company called Celera, and the other was a publicly funded group. Now, what's important in this is that at that time, nobody really knew what this meant. It was a hell of an achievement. Everybody agreed to that. But what could you do with this? And the challenge really was, so in each of these years, you see these many nature papers that, nature and science papers that are published here. Every time something groundbreaking was achieved, people would try and postulate, what could you use this technology for? But it really wasn't clear, right? And so it's sort of like when Wi-Fi was first discovered, people really didn't know what it could do. Today, now you and I can't live without Wi-Fi, right? So, so at each of these junctures, each of these years, something new would come up. They do a new chromosome, or they do a new animal model, or some such thing, and eventually they publish the sequence. So you can imagine now, right? $2.7 billion and 15 years. What do we do next? All right. And that's when the disruption came in. So during this time, there were two reasons why it finished ahead of schedule and below budget. The main reason was sequencing technologies were developing and evolving during this time, right? So 1973 or 75, Fred Sanger got a Nobel Prize for discovering Sanger sequencing. That is, he pioneered the way in which DNA was sequenced. And of course, in those days, and it wasn't long ago, because I was doing my PhD in 95 to 2000, and I used Sanger sequencing methods in the background of what was going on in the whole genome sequencing project, right? So it wasn't that primitive, I'd like to believe. And it was a very effective way in which to organize the nucleotides in a genome. But it was slow and very time consuming. And therefore, people could probably hope to sequence one gene or two genes in their entire PhD in their 5 years. Okay, and even as this was happening, the technology around sequencing was improving in spades. So what happened next was disruption. So I'll just follow this chart. You look at this green line, right? So we're looking at well over $100 million in 2001 was the cost per genome, right? And as you track this down, it was going down fairly steadily till 2007, right? And then there was a sudden crash in cost per genome, all the way down in 2015, when a company called Illumina announced that the $1,000 genome was now available. In other words, what cost $2.7 billion and took 13 years could now take 3 days and cost you $1,000. So this sort of sharp improvements in sequencing technology are very often plotted against Moore's law. Okay, now Moore's law is not any solid theoretical physics law or anything like that. It's more of an observational law that gives you a kind of handle on how disruptive your technology is going to be. So the classic example is the transistors in an integrated circuit. So people who are aware of this in the semiconductor and the computer industry know that practically every year the number of transistors that they can add into the circuit doubles, right? And thereby creating the computational improvements and ameliorations that we see in computation and technology throughout our lives, right? So if you were to track that, Moore's law is this white line over here. So if you compare this green line to the white line, you can see that in terms of disruption, the genomics industry and the cost for sequencing a genome had far outdone what the semiconductor industry and the computer industry was doing. So just to give you an idea that companies like Intel always thought that Moore's law was the absolute limit at which technological improvement could take place. Interestingly, why that happened was, and why you see those sudden inflections, was around the year 2005 2005, 2006, a company called Roche, right? It's actually a big pharma company called Hoffmann La Roche, makes many, many drugs, but they're a very diversified conglomerate. And so they came up with a technology that could sequence DNA, right? And it could sequence it in what they called a massively parallel, high throughput manner. So the difference between the technologies that were used before, the ABI technologies, this used Sanger sequencing, mind you. This used a very similar principle to Sanger sequencing in that the biochemistry was the same, right? But they did it in such a massively parallel manner, and the throughput had to be so high that you could get a lot of data with one run, and therefore you shorten the time in which you actually established your genome, right? It was soon followed up by the acquisition of a company called Solexa by another large American company called Illumina that bought this technology. And from that point onwards, the entire crash in the pricing came almost exclusively from the technology that was being developed by Illumina, right? And so to make a long story short, what did they do? They could sequence far more DNA because they were doing it parallelly, and they did it in a shorter time because it was being done parallelly. All right. I'll talk a little bit about the biochemistry behind it in the next couple of slides, but one more time to just pause and say that if you get lost in the next slide, don't worry about it. It's just that more DNA could be sequenced faster because the technology had improved. This is a slide from the company Illumina, right? And this is a 2-year-old slide or 3-year-old slide, and already these top 2 machines are obsolete. In other words, there's technologies that are even better than that now available. Okay, so all I'm saying is that if you blink for a second, a new machine comes out, a new technology comes out, and you'll be left behind. To give you some idea, this machine may cost, it's called a MySeq. It'll give you about 15 gigabases. Gigabases, remember I told you there are 4 bases ATGC? So gigabases, 10 raised to 12 minus 12, or 10 raised to plus 12, right? And so you have gigabases, 15 gigabases of DNA here, right? That generated in one run. And you go all the way in this subset to close to 1800 gigabases, okay? Which means you can do terabases when you do more than one run, right? Because these are groups of 10 machines. So like I said, these 2 machines are already obsolete. You had to buy these in groups of 10 and 5. So the X10 and the X5 had to be bought in groups of 10 and 5. And just to give you an idea of pricing, this machine costs $100,000, give or take. And this machine over here in the middle costs about $1 million. This one is 5 times of this machine, so it's $5 million, and this was 10 times this. So it's $10 million. We're talking 74 crores or 75 crores for this, right? Versus 70 lakhs for this. So as the price range varies, it varies as per the amount of output of gigabases that you can generate from that machine. So it's like having an auto rickshaw to transport people, and a Volvo bus to transport people, and an entire train to transport people. And so the per sample cost keeps coming down because you're doing so many more samples. So it's just a fraction. The reduction in cost is just a function of the increased scale of output that these machines are capable of generating. This machine can generate in 28 hours. It can generate 1.8 terabases of DNA, which is shockingly large, and is the benchmark, set the benchmark for the average cost of a single human genome to be sequenced to be $1,000. Okay, so I hope this is a good point to stop and get technical. These aspects are very important because remember I said to be a disruptive technology, you have to have accessibility. How many people have access to $10 million to set and invest in this? You won't know till you look. And while I was in this company in Illumina, we actually sold this machine at $10 million to a company in India, because they see the huge value that it might bring in the clinical genomics domain. All right. I'm still very much in touch with them, and I'll mention them in a few slides coming up. It'll be a very interesting company for those of you who are interested to follow up with. Okay. But just when you think that these things are out of reach, they are very much within reach. Now that somebody has invested the $10 million, you and I can simply go to them and pay $1,000 and get our genome sequenced. Okay. Why we need to do that, I'll come to in a few minutes. Before that, let's see how. Okay. Again, this is a good time to pause, because I thought I should put together a little layout of what lies ahead. I think we're doing okay for time. So basically, I'll talk about the sequencing methodology. This is going to be a little technical in terms of the biochemistry that is used, as well as some of the technology that is used. I'll give you a sort of detailed example of what first generation sequencing, second generation sequencing, and third generation sequencing are. These, just for your information, first generation sequencing is just referred to as sequencing. Second generation sequencing is referred to as the next generation sequencing, and third generation sequencing is something that is very new and current, and I'll only briefly touch upon it. Because it is an evolving technology. It is not by any means the same price and therefore the same accessibility for all of us, yet it has certain advantages. It a far more attractive technology to have. But for the most part today, when people talk next gen sequencing technology, they talk about a company called Illumina and their technology. Okay, it's called sequencing by synthesis. I'll then briefly talk about the applications, and this is probably a little more interesting and less technical, of how people have used this in cancer, reproductive health, and infectious disease. And I'll briefly tell you all the other areas in which it can be used, but I'm not going to talk about because we don't have enough time today to talk about. But part of what I wanted to impress you with is that it's finding its way into every niche, and by no means have we covered every niche. So some of you today who are listening to this may actually find a utility in something that it hasn't been used for before, and that becomes the next niche in which you can use sequencing. So it's a brand new technology. Please explore it and find out as much as you can, even after this talk is finished. Finally, this is, I thought, something I should put in there. It's a slide on the sequencing opportunity landscape, primarily in India. Because there are a large number of students here, I thought, or I expected a large number of students here, I thought it would be wise to put some names of organizations out there and their sort of classification, so that in case one of you wanted to actually read further or actually work with them or join them, you can reach out to them and try and see if you can intern with them, or once all the COVID social distancing era is finished. I will highlight in red which part I am on. So if you lose me at one of these parts, you will get back to me at the next red part. Okay, and let me begin. All right. So the first sequencing methodology that I'll talk about is called Sanger sequencing. Okay, like I mentioned earlier, Frederick Sanger got the Nobel Prize for discovering this form of sequencing. And you will see that most of the other technologies I talk about, including the one from Illumina, the next gen sequencing, second generation sequencing, uses the same philosophy as this method. And that's why I thought it's important to go over it. This may be something many of you already know, but I'll talk about it briefly, right? Basically, a sequence of DNA that needs to be, or a piece of DNA that needs to be sequenced, and by sequence I mean have its bases organized. What is done is you first clone it, right? You clone it into a vector and you make many copies of it, and then you add a combination of dideoxynucleotides, and you add a DNA polymerase. And DNA polymerase is an enzyme that, based on the presence of a primer that you can see here, will anneal to its complementary strand. And if a DNA polymerase is here, it will see this primer and will start elongating from a 5' to 3' manner. So what Fred Sanger did was he added a certain proportion of what he called deoxynucleotides, right? Dideoxynucleotides. They're called DDNTPs. Effectively, it's just like a regular DNTP, except there's this OH is not here at the 3' end. So this chain cannot elongate, right? Now he added them in a proportion such that at varying lengths, eventually one of these DDNTPs would be added, and it would block chain elongation from taking place. And then, of course, he ran electrophoresis gel and separated the different size fragments out. And then in his case, he used radioactivity to detect the different sizes, and then he reconstructed the actual DNA sequence. But a slightly more modern method actually incorporated differently labeled nucleotides. So the TTP was red in color. So they added fluorophores. CTP was blue, ATP was green, and so on. And so when they fired a laser through this capillary, they could tell which nucleotide was added, and then they could reconstruct the entire DNA sequence based on this arrangement of colors that came out as a result of running this capillary. Okay, so I won't go into too many details. Suffice to say, two important things here. Dideoxynucleotides were required to stop the reaction at certain places, and a laser was used to excite the fluorophores and capture the exact base that was present in that place, right? And that kind of theme continues. So we'll go on to the next one, which is Illumina's technology. This is what most people call next-gen sequencing, right? And this is what caused the big revolution, the big inflection point in terms of how much DNA could be sequenced at a time, and the concomitant drop in prices, right? So it becomes a very important thing, right? The entire workflow consists of, I guess, 3 main steps. Okay, the first step is to prepare a DNA library. So you take the piece of DNA that you want sequenced, and you construct a DNA library. In other words, you add adapters to it. So you take genomic DNA, you break that DNA into smaller fragments, and you add certain linkers to it. Okay, so this is by a standard ligation reaction. At this point of time, each one of these fragments, if rearranged back in its original manner, would give back your entire genomic DNA, which you see is this twisted lump here, right? Now, the next step was to process it and put it using microfluidics, which, to the best of my knowledge, all you physicists are very good with. So you see this little glass slide, and you see these thin lanes in here. So in these lanes, actually, they have coated DNA strands that are complementary to the adapters that were added here, right? And using microfluidics, they flow all these library molecules through these lanes, right? And as they flow through, since they're single-stranded, they find their complementary strand, which is on the adapter, and they go and bind, right? Once they bind there is a small in situ PCR reaction that is conducted there. About 10 or 12 cycles of PCR are conducted there, where you want to amplify that strand, and there's something called cluster formation and bridge amplification that takes place here. I won't go into the details, but suffice to say, you increase the number of representations of each one of these when it's on the slide, right? And the last step, so this is done on a separate machine, okay? It's called a CBOT. And then taken and put into a sequencing machine, to which DNA polymerase is added. And of course, there's an elongation reaction that takes place, very much like in the earlier Sanger sequencing method. So as each color gets added, right, a picture of the lane is taken, right, and the laser is fired, and the excitation results in, depending on which nucleotide is added there, the excitation sort of results in a color of a particular nucleotide, right? And this is done at every cycle, right? So at every cycle, the DNA polymerase adds a nucleotide. This cycle is stopped because it has a base termination nucleotide that's added there with a certain color. Those that have, an A will have a certain color, C will have a certain color, so on. And this entire grid, this entire region that you see here is sort of geospatially marked out, okay? Now you can see the size of those dots, right? That's because these are clusters. Three main technologies came together here. Of course, the biochemistry of Sanger, because it's the same thing that they're doing here, right? The fluorophore technology that that ABI Prism had, ABI group had brought into picture, and the excitation of those fluorophores to give different color spectrums, but also CCD camera technology that could resolve two spots at this distance. Okay, so it wasn't just biologists who got together and made this machine. There's a whole bunch of engineers and physicists who put all their physics and engineering knowledge into it to try and build this. And of course, after this kind of data was generated and pushed into a computer, it was very important that there was a co-evolution of high-end computing that took place. Okay, so back in 2010, I think having 24 processors and 48 parallel processors was considered a big deal. Today, I think anyone can kind of assemble a server like that at home, right? And of course, the computation requirements have grown even higher and higher in sequencing. So at various levels, many different technologies have come together, right, to generate sequence information. So this is not by any means restricted to biologists. This crosses over into all areas, right? Once, of course, you get that sequence information, I won't go into the details here, but you can do two things, right? If you already have a reference sequence, so remember I mentioned earlier that the human genome has already been sequenced. Now that it has already been sequenced, it's referred to as the reference sequence. So if ever you or I sequence our DNA today, we align the sequences that we get against that reference sequence. And so it makes the reassembly of our DNA very easy to do, okay? Computationally very easy to do, because you're just simply doing sort of blast kind of alignments back with a reference database. It's a little more challenging when you're trying to sort of construct or reconstruct a de novo genome. In other words, maybe there's an organism that's very interesting that you want to study, and you think there's something interesting about the way its DNA is organized. Then it's a little more difficult. So you have to do far deeper number of reads. You have to do different read lengths. You have to build a scaffold. And the process of assembly is a slightly different process, and a slightly different statistical algorithms are applied. In both cases, there is a requirement for overlapping fragments of DNA in order to construct scaffolds and contigs, and then put together finally the entire piece together finally the entire genome for your perusal, as the case may be. So if you got nothing out of this, on the left side is resequencing, where you just do alignments. So if there's a reference sequence available, you use a certain set of protocols. If on the right side is if you have no idea what this organism's genome is like, or its nearest ancestor is like, well, then you use a completely different set of algorithms to reassemble the genome in a de novo manner. All right. Now, just to make things more complicated, because it's never good to have a monopoly, and Illumina has a monopoly in next generation sequencing, other technologies also started showing up. So I won't go into the details here, but while Illumina uses fluorophores to detect new nucleotides added, this particular company, Thermo Fisher, uses the ionic strength. So every time a new base is added to the DNA strand, then the ionic strength of the solution changes. And so this is literally a semiconductor chip that you see here. It's one large semiconductor, right, with these little millions of sensors in each of these wells. And in each one of these wells is a molecule of DNA that's being synthesized. And as each nucleotide is added, the pH or the ionic strength of that solution around it changes. A, T, G, and C, as a result of which you can reconstruct once again what the genome sequence is, right? And sort of bioinformatic methods are very similar to the one that I mentioned before. So assembly and resequencing are the same. Once you have the sequence information, everyone sort of converges back into the same sort of pipelines, bioinformatic pipelines. There's another company called BGI, which is a Chinese company, stands for Beijing Genomics Institute. They bought an American company called Complete Genomics, and they use a technology called DNA nanoball sequencing. Once again, I won't go into it because I may have lost many of you already. The point I want to make here is there are different technologies, and each one comes at this problem with in a slightly different manner. But in each case here, all of them have some kind of termination nucleotide to help them in sort of creating different strand lengths that eventually they assess for the terminal nucleotide. Okay. Now, this is the last technology that I'll talk about today that exists out there. It's called the third generation. So if if I go back, the technology I mentioned about Illumina is the technology that has brought the price down to $1,000 per genome. Okay, and BGI has pushed that envelope further and brought it to $600 per genome, and everyone is fighting to see who can bring it to $100 per genome in the end. And eventually somebody will do that, and then eventually it'll become $10. And as that happens, more of us will have access to it, and as it gets more accessible to us, we'll have more uses for it. Some of those uses I'll talk about in the application, but here is a technology that approaches this completely differently, right? And these guys on the left, it's a company called PacBio, Pacific Biosciences, right? They use a unique method in which they have these zero mode waveguides, right? These are little well-like things where they've immobilized, DNA polymerase, and they take large molecules of DNA and pass it through the strand. And there's a videography nucleotide is added, very similar in terms of addition of fluorophores, and there's a reconstruction via video of the sequence that they actually try and achieve from this. But the important thing here is, unlike the others, which are called short read sequences, never exceed 300 to 500 bases, right? In this case, you can sequence up to 80 kilobases of DNA. Right? And the reason for wanting to sequence longer pieces of DNA is because in many eukaryotic genomes, you find the presence of repetitive elements of DNA. Okay, and these become very challenging to sequence when you do short fragments, because everything is an alignment process. And so if you have 80, 80, 80, 80, 80 repeated many times, you don't know which 80 comes where, right? So it becomes a bit of a challenge to align those and reproducibly and get a good result. These long molecules, or these long molecule sequencing technologies, go up to 80 kb of DNA, right? So the actual repeat sequences that are present well within that 80 kb region, and so you can get very accurate, sort of structural knowledge of the DNA, right? So you're much more sure that this DNA has X number of repeats as compared to the short read technologies. And short read people have used bioinformatic methods to work on that. I mean, that race continues. But the fact is, these are very interesting technologies. Right now, they cost about 10 times the cost. So this would, for every $1,000 genome there, this would be about $10,000 to $15,000, right? But you can find some very cool and interesting aspects of this. There are no artifacts of PCR and so on and so forth that come from this, and that we can talk about in a more technical discussion later. The coolest technology available today is Oxford nanopore technology. Again, it's a long range thing, and the most recent paper sequenced a 2 megabase section of DNA. It's that long, okay? And the utility of that is just phenomenal. However, the error rates of the sequencing, it actually uses a protein nanopore across an artificial membrane, and there's a current applied across this membrane. And as each nucleotide goes through that pore, you get a change in the current, and that corresponds to the sequence. Unfortunately, it's still rather error prone and cannot be used in the clinic on its own. It can be used in combination with other technologies. But the coolest thing about this technology is it's portable. It's about the size of one of those large iPhones, not the regular size ones, the sort of slightly bigger ones. That's the size of the entire sequencer. So it's extremely portable, and if you want to do sort of field studies, you can do it right away. Like I said, error prone and a little expensive at this point. So we'll leave it at that. Okay. That's just a bird's eye view of the sequencing technologies that exist out there. I'm going to rush through some of the applications. Most of you may know these applications, and I'll try and do this in the next 5 minutes, and then we can quickly run on. Okay, so the universe that's impacted by genome sequencing, quite frankly, I don't think there's anything left out. In agriculture, one can use genomics. Biopharma, people use genomics. Cancer, people use genomics. Complex diseases like diabetes, people are beginning to very cool and interesting thing. The kind of thing you'll get if you go to a gym and they'll say you have to eat this food based on your genomic content. So it's a more of a fun genomics right now, but I think eventually will become very important to how we run our lives, given the advent of wearables and other things that sort of can work with genomics to understand health and wellness better. Forensics is probably the most ancient use of genomics, where most of us are aware of. I won't go into that at all. Genetic health is something what my company TGB Diagnostics does, right? Where we look at families that have genetic disorders in them, and we try and elucidate what are the reasons and rationale behind it. Infectious disease, I'll talk a bit about it in this. Population genetics, this is very interesting to read because you find that many populations are and can be resistant to certain types of infections. I think it's too early to say, but I believe that India has a very low mortality rate for COVID compared to other countries. So population genomics would be a wonderful thing to use to try and understand what is unique about our population structures or genomic structures. I won't talk about it, unfortunately, today. Reproductive health, I will talk a bit more about this, and of course in research work. Okay, so this is the current landscape of where all genomics has been used. I cannot talk about all of them. I'll run through 3 of them in the form of examples. The example in cancer is, what do we need in cancer today? In cancer diagnostics today, you need, the goal should be at one level, you want to assess risk, right? So a patient arrives, and what is the risk for that patient to survive this disease, and then stratify them according to that. So you may have heard of stage 1, stage 2, stage 3 types of cancers, so on and so forth. Another important thing is screening and early detection. Okay, screening becomes a very important aspect of understanding whether a person is going to get cancer or not. And it's still kind of in a very immature stage, which makes early detection very difficult. I'll show you in a slide later how important early detection is. Of course, you want diagnosis and characterization. You want to know which genes are mutated. Make no mistake, cancer is a genetic disorder. It's mutations in the genes that result in cancer and tumors. So it's almost directly related to genome sequencing as a technology, right? So if you sequence the tumor, you can diagnose what genes are mutated and thereby characterize it. Of course, you have prognosis. Prognosis is kind of based on this diagnosis, you need to know what to do with the patient, what kind of therapy to prescribe, so on and so forth. And an interesting area called theragnostics, right, where it's a combination of diagnostics and therapy, right? So as you're treating a patient with a certain type of, let's say, regimen, you treat a cancer patient with a certain type of regimen, you keep testing and sampling that tumor to see if those tumor markers, based on the mutations you're seeing, are actually reducing or not. If they are not, you can then intervene and change the therapeutic regimen. Okay, so that's theragnostics. And of course, monitoring. So once somebody has recovered from cancer, or maybe somebody doesn't have cancer but needs to be monitored, that's another area where diagnostics in oncology would be very useful. And genome sequencing kind of fulfills all of these. Okay, I'll talk about only a few of them. When tumors from, say, the lung, a lung adenocarcinoma, lung squamous cancer, breast cancer, colorectal cancer, melanoma, or head and neck squamous cancer, when all these cancers, when the tumor was taken and sequenced, the DNA from the tumor was taken and sequenced, many genes were seen to be mutated. Okay, now traditionally what people would do is, okay, or a clinician would do is, there's a regimen as prescribed by, let's say, the NCCN guidelines that they follow, right? That lung adenocarcinoma has to be treated with XYZ kind of therapeutic protocol, right? And if it's a breast cancer, it's a different set of chemotherapeutic regimen or whatever, targeted regimen, whatever it is. But each one of these were defined by the origin, the tissue of origin. So the cancer, wherever it originated, that tissue decided what the therapy would be. This is how it was in the good old days. But because of the ability and access of sequencing, today, if a person has cancer and they sequence the tumor, they can actually list out which genes are mutated within that particular person's tumor. And it's seen that many of these genes are actually mutated across in different people who suffer from different other types of cancer. And so eventually now, where the clinical world is sort of conflating is, we have KRAS inhibitors, by the way, we have NRAS inhibitors, right? So why not use them? You may have used them in lung adenocarcinomas in the past. Why not use them in melanomas going forward, right? And so now we are beginning to reclassify cancers as by the genes which are mutated in them, and thereby prescribing therapies according to that. Now, this is a huge revolution in how cancer treatment is actually being approached, and it's all thanks to the fact that we can now easily sequence people's cancers to understand the underlying sort of genetic mutation that is the cause for that cancer developing, right? This is another kind of important thing. So when you want to, there's a strong correlation between early diagnosis. So let's go up here. So in lung cancer, right? If the number of people who are diagnosed at very early stage are only 15%, and at late stage are 57%. Conversely, the 5-year relative survival is very high in the early stage if detected early, but horrible the later it goes, right? So what can we do in order, is there a technology that will help us pick up lung cancers earlier than they are currently being picked up? Something very similar for ovarian cancer, right? So you can see this 5-year survival curve that just sort of plummets by the time you're in stage 4, right? And it becomes really important that the take-home message is it's important to detect cancer early, because if you detect it early, you can cure cancer. It is curable, right? And so there is a company called Grail, right? It's named after the Holy Grail from the Bible, because it is really the Holy Grail of early detection in cancer. This is a technology called liquid biopsy. So cancer cells are known to, because of apoptosis, are known to shed their DNA into the bloodstream of the host. And it's easy to detect, not easy, it is possible to detect the cancer, the tumor DNA, and separate it from the rest of the sort of free-floating DNA that other normal apoptosing cells leave behind in the system, right? And those, so imagine in the future, you go for your annual physical and you do a blood test. Today, you can do your HbA1c for diabetes or whatever other indication there is. But tomorrow, you'll be able to actually see if you can detect an early sign of cancer, right? Because this can come from your blood. Now, this technology is not yet there, right? But companies like Grail, $200 million, $1 billion valuations, $5 billion valuations, they're all unicorns, right? These companies are working very hard to draw tight correlation between doing a liquid biopsy and detecting early cancer. And if this actually happens, and I predict it will in the next 2 to 3 years, right? It's going to change the way we detect cancer in terms of how early we do it. And conversely, it's going to have an equally important role to play in the survival of the people who are sufferers of cancer. Finally, one last thing I'll talk about with cancer is something called tumor mutational burden. Without going into the details, the more the mutational complexity, or the higher the number of mutations there are in a tumor, the more amenable a patient, especially with lung cancer, the more amenable a patient of lung cancer is to what is called immunotherapy, right? So these are checkpoint inhibitors, right? So the tumor cells have figured a way in which to fool the immune cells that would normally recognize them as tumor cells and kill them. These tumor cells have figured a way in which to fool those cells using a protein called PDL1. It binds to the activated T cell, right, which is part of the immune system, and prevents it from mounting a response to kill it, right? So this clever strategy has been figured out by the tumor cell. As a human race, we're equally clever. We figured this out, and so there's a molecule called Keytruda, which actually inhibits this interaction between these two proteins and resensitizes the immune system to attack and kill the tumor. And it's a very, very high, very, very good prognosis type of therapy. Okay. However, in order for patients to benefit from this, it's important that their tumors are extremely complex. And therefore, what people do before they send such a patient for immunotherapy is they sequence their tumor DNA. And once they sequence the tumor DNA, this is an index on which they rank it as being high TMB. If it's high TMB, then such a cancer patient will have a very good prognosis if put onto immunotherapy. Okay. Sorry, I have to rush through these slides. They're interesting as they are. Sequencing is also used in what is called non-invasive prenatal testing. Once again, I'm going to rush through this. Similar to cancer cells that apoptose and shed their DNA into the bloodstream of the host, so also does the fetus. So in a pregnant mom, the fetus will have a set of apoptosing cells that shed their DNA that goes straight into mom's blood circulation. Okay, so a pregnant lady can do a blood draw around 10 to 12 weeks after conception, and we can detect fetal DNA. Not only that, using an interesting counting method, which I'm not going to have the time to discuss, one can actually decide whether there is an aneuploid chromosome. An aneuploid chromosome is a chromosome that is present in higher number than required, right? Which would be indicative of something like, for example, Down syndrome, right? 21X trisomy, right? So these are features that can be detected from just doing a blood draw, right? And so maternal blood is known to contain anywhere between 2 to 20% of cell-free DNA, which is fetal in origin, okay? And so the idea is if you maximize that, you can detect for the ploidy in the fetus and thereby detect Down syndrome. The important thing here is even though it requires a blood draw, it's non-invasive. Many of you may laugh and say, no, 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 as long as there's a needle involved, it's invasive. Let me tell you what people use in lieu of this, right? So before this technology came up, you see these needles, these massive big needles were actually run into in a methodology called amniocentosis, and the cells were taken out and then stained for ploidy using karyotyping. Okay, so just imagine one of these needles going in. It's not pretty at all. So compared to this, NIPTs are non-invasive, right? These sort of traditional technologies also increase the risk of miscarriage because of the technology itself, right? Pushing that needle in, and so therefore it also comes in the way of a healthy live birth, right? We'll skip through this, right? Effectively, it's a counting protocol, and if I can get to my last slide here. What they do is basically there is some proportion of fetal DNA and there's some proportion of maternal DNA. And if you have so in all these cases, it's, you see an even number here, but in the case of the trisomy 21, you see an extra sort of signal coming from the sequences on this chromosome, as a result of which you call it an aneuploidy. And of course, at that point of time, at 10 weeks to 12 weeks, you can take a call whether you want to continue the pregnancy, terminate the pregnancy, or in any case, whatever your path is, you are prepared that your child may or may not have 21X trisomy, right? So that's the idea behind doing these kinds of screenings. Now, why this is important is, as urbanization increases and all of us are running around getting jobs, there's a general trend that's being noted that women are having their babies later and later and later in life. So after 35 years, women who get pregnant, I mean, this is the first pregnancy, if they get pregnant, then it's called something called a high risk pregnancy, which means the chance of trisomies is very high. And so this was almost a mandated testing protocol in high risk pregnancies. But nowadays, even younger mothers are using it just to rule out anything that could go wrong, right? So I think now it's being advised for high and mid-risk pregnancies, but I think even low-risk pregnancies will soon start doing it because it gives you greater confidence in what testing that you're doing. Finally, sequencing is used in detection of infectious diseases, and this is My colleague, Dr. Nikhil Phadke from GenePath in Pune, he talked about this case, and I've stolen this slide from him. Basically, a patient presented themselves with an eye infection that could eventually risk blindness. Now, in a traditional world, it could be a bacterium, virus, or fungus, and each of these has a different kind of sort of therapeutic agent: antibacterial, antifungal, and antivirals, right? And some of them are contraindicators of each other. So it'd be very difficult. They could actually endanger the life of the person if all three were given. So what is typically done is a swab of this eye is taken and cultured on a plate, and then after 5 days, that culture is tested for various features, and then of course whether it's the bacteria, so on and so forth. In this case, this patient comes in, he takes a swab, he just sequences the DNA in that. They discovered it was a bacterium, right? This is in a matter of 16 hours, right? They identified the bacteria as Stenotropomonas, and it's a known ocular pathogen, and it was easy enough to find an antibiotic that they could use to prevent it. And it was a very simple case in which genome sequencing came to the rescue. A more popular one that we are all talking about now is COVID. Many people don't understand why sequencing COVID-19 was a big deal because we're all still stuck indoors. None of us are getting to move out. But let me give you a few reasons why. So if you concentrate on the right side, right? The fact that COVID-19 was a virus, a coronavirus in itself, and the identification of that virus and what it was from Wuhan was only because the sample was sequenced. Okay, and next-gen sequencing technologies were the technologies used. All the genes that code for the viral genes were actually sequenced. So we know the protein and the RNA and the protein that comes out of this, right? We could immediately tell that it's a closer relative of SARS and MERS, and it's from the family of coronaviruses, right? We also could identify that it came, what the animal vector was, because we look at the nearest relative and we found out it's a bat virus, right? And that was available in the database. So that's how you know it came from bats, right? Sequencing will help track the viral. I know there are many conspiracy theories about that. We can leave that out for the time being. But sequencing also helps track the viral spread, as done by Vinod Skaria and team in IGIB. And they've drawn this cladogram where they show that in India, 5 different clades of this virus are there. Clades simply mean small mutations that can then be tracked and then paid. So let's say at this particular node, a mutation takes place, right? And then each of these nodes, a small mutation this way, that way is taking place till eventually it's so different from each other that this entire thing is different from this. So they identified 5 different clades in India. And this is interesting because you can actually track. He told me this one, the B that ends over here. So this is the time axis. This particular one is the Wuhan virus. So the first set of patients who traveled from Wuhan and came here, this was the sequence that they had, and see the variations that have taken place and how we can track this going forward. Most importantly, because of the sequencing, protein, antigen, and mRNA-based vaccine therapy trials could start way back early in January itself. So many of you are wondering how by September, something that normally takes 5 years, 6 years to bring out, something is already in phase 2 trials. That's because they had a head start on understanding what this virus was, what antigens that they can use for vaccination. And so the fact that we're closer than ever to getting a vaccine, I hear the September, October is a good time to expect the vaccine. We'll either know it's worked or not, after which we'll have to do a lot of other things. So with that, I'm going to stop the stuff that sequencing can do and basically tell the students who are on this call that when I was in this field in 2015, I was the only lab that actually did this kind of sequencing as a service provider, right? So people could come and give their samples to me. I was the first people to establish it as a for service tool, 2015. Today, that landscape has changed so dramatically because of the opportunity that this presents. So those of you who are keen and interested in this technology and learning more about it, you will be happy to note that there are many genomics companies in India. Of course, you have the technology companies, I think outside of Thermo Fisher Scientific, none of these actually has a presence in India. They have distributors. So if you're looking for a job, or you're looking for experience, or you're looking to intern, you can find out who the distributors of these technologies are for these companies, and you can work with them. And it's actually very, very fascinating and interesting experience working with them. So those companies I can always sort of tell you if you're interested in later. And these are the companies that actually work the technology, right? So who builds the machine, who builds the biochemistry, all that. These are the guys. Very interesting to be part of this. All the work goes on in the US. so you'll basically be doing applications over here. The service providers are those people like Strand, MedGenome, Genotypic, and Sandor. And all these guys actually will take a sample from you, provide your sequence information to you based on whatever you need, and generate a report for you. Okay. So this, if you're doing research work, you can come to them. And you could also go to research labs. I think IGIB in New Delhi has done a lot of work on COVID specifically, but other diseases as well. IISc Bangalore has, NCBS has, and of course CCMB in Hyderabad has. Many of these places actually have. There are many more that have. I'm just highlighting a few here to start your searches if you'd like to learn more about it. Finally, most importantly, the clinical labs like GenePath TX, Eurofins, Strand, and MedGenome. In the case of clinical conditions that you may have, you can come to them, or any hospital you go to, you can get these guys to come to the hospital and collect your sample and take your diagnostic characterization further. So without saying anything else, let me thank you one more time for your patient ear. I look forward to hearing some questions at least. Just to let you know, the future is already here. This is not space age discussion. It's just not evenly distributed, and that's why I gave you those names of places. It's a good place to start your your explorations on learning more about this. Thank you.
Yeah, thank you. Actually, one participant had raised a question.
Yes.
He had said the future seems so exciting with the intervention of the CRISPR gene tech. Yes. Apparently, future relies working in a supercomputer with CRISPR sequencing. Yes. Share some of your views on it, sir.
Yeah. Look. So if today belongs to the era of genomic sequencing, right? Once you have all the sequence information, the question really comes down to, excuse me, what can you do with it? And that's where CRISPR-Cas methodologies are really, really vital and important. Okay, so those are the technologies that once you know the sequence towards which you want to make a modification, you can actually go in and make a genetic modification. I mean, there are many companies, I think in the US, Editas and a few others, that are actually doing this, right? So they're going in and correcting errors that are there in the genome. Definitely very exciting, but to be very honest with you, it's not there yet. Okay, gene therapy is a bit of a promise that has been waiting to happen for some time now, and CRISPR-Cas is the best chance we have at delivering gene therapy. But we have to wait a little. It's not just the functioning, efficient functioning of the system. Many of you may have read about a Chinese scientist who actually genetically modified embryos. So the story goes where he modified the CCN5 gene, which, by the way, if it's a wild type gene, then a propensity to get HIV is high. So he mutated those genes, and so the fetus and the babies that were born thereafter had the mutation and will be immune to HIV infection, right? While this was, groundbreaking discovery and announcement in Hong Kong, like many scientists who make these kinds of huge leaps, he has disappeared. Okay, and the reason for this is the world doesn't seem to be ready to accept, ethically speaking, the intervention of genetics. It will take time. It is the regulatory framework that has to be built around it that will build confidence. But I think the worst case scenario is tomorrow, if you only want to have blonde hair, blue-eyed babies, it can be done. right, using this technology, and that will, what does that speak for the diversity of humanity, so on and so forth. So it's a controversial point. I think once the regulatory environment fixes some parameters around it, I think we're likely in the next decade or so to actually see it playing a role in cancer and other diseases that I mentioned here, specifically from the point of view of correcting genetic errors. Good question. Thanks.
Another question, what about Maxim Gilbert's tech?
You know what? It's just one of those things. I don't know if you existed in the era before Google. Okay, we used to have search engines called Magellan. We used to have a search engine called Alta Vista. AOL. There were many search engines, and all of them had wonderfully written algorithms. But for some reason, Google took over, right? And today, Google is that technology. Now, if you ask me, why did the others not do it? I think even Yahoo had a search engine. It is not that their algorithm was any inferior, or this Boolean statistics that they use was any different, right? Some things are just, it just so happens that way. And I think in the case of Maxim Gilbert sequencing, that's really what happened. It seemed to have a limitation in terms of how many people sort of adopted it, and Sanger didn't. And therefore, that's the path that it took. Good question.
That's all regarding it, because okay. I think a few students are mostly from physics background. One is, which field is good for research, either biological sciences or biotech? Biological sciences
biotech or biotech, is it? They're very similar fields. Possibly, are you talking about bioinformatics and computational analytics, maybe? Because that seems to be something that's very hot. It's really based on your skill set, right? I can tell you this much. It's no longer useful knowing one thing very well. If you're I think the best way to illustrate this is, I was at a very large meeting in genomics recently, and the quite world-renowned scientist Partha Majumdar, he's the ex-director of NIBMG, was giving a talk, and the audience was filled with clinicians, primarily pathologists, right? And he was saying, that all these benefits of genome sequencing, so on and so forth. And one of these clinicians sort of cynically stood up and said, you may say all these things, but there's nothing better than a propydium iodide stain and EGFR mutation check. And he seemed to write off the entire talk. And Dr. Partha turned to him and said, unless you adapt now, in 20 years, you may not have a job. Because that's how fast fields are evolving now. So don't stay stuck to one particular field. It's good to have, at least at a younger sort of developmental age, maybe in your bachelor's, master's, even to your PhD, to have some kind of grounding in biological sciences. Because once that grounding is there, you can always explore other areas. And if you have an interest in computation, you should take it up. I think the world in the future belongs to generalists and multitaskers. And I don't mean jack of all trades, I mean master of all trades. So you have to work very hard towards knowing as many disciplines as possible. And of course, there's always going to be place for the pure science disciplines. But I don't think that answers your question directly. The direct answer is, do the one that you are happier doing. You'll go further doing that.